Welcome to the video for chapter 13.6. This is the last video of Unit 5 before the Unit 5 exam. So in 13.5, we talked about mechanisms of action for antibiotics, and we started to touch on antibiotic resistance and some of the ways that bacteria can gain resistance at the end. Well, in chapter 13.6, we're going to talk about all the different ways that antibiotics can be resistant. Okay, so it's called the challenge of antibiotic resistance. Before we jump into the material for this section, let's do a review question. Now, this is what I would call a critical thinking review question. This is going to take some of your knowledge from the last section and some from the previous chapters that we've talked about in Unit 5, and it's going to ask you to kind of put them all together. So review question number 1 says, hypothesize why there are more chemotherapeutic agents that block bacterial protein synthesis than chemotherapeutic agents that block viral protein synthesis. Okay, so A is viral protein synthesis occurs inside the host cell while bacterial protein synthesis does not. B, viral protein synthesis relies entirely on host ribosomes. C, bacterial protein synthesis relies entirely on host ribosomes. D, viral protein synthesis does not involve ribosomes. Okay, so take a minute, pause the video, look at your notes, and see if you can't find the answer. Okay, so let's walk through this question before I tell you the answer. There is one of these, A, B, C, and D, that we can automatically eliminate just based on what it's about. Okay, so it's talking about protein synthesis. What organelle does protein synthesis, guys? The ribosome. Okay, you always have to have ribosomes to make proteins. D is incorrect because all protein synthesis involves ribosomes, whether it's bacterial, viral, eukaryotic, we need to have ribosomes. So D is automatically wrong. So this cuts down one question, so we're left with 3. Now, for the rest of these, we kind of have to think about the difference between bacteria and viruses. What's one of the biggest differences we have between these two? Well, the first one that comes to mind is viruses are non-living, right? They have some characteristics that they have some characteristics of life, but there's a lot that they don't have, right? They can't reproduce on their own. They don't have that molecular machinery. They are a genome in a protein jacket. Okay? So viruses, in order to make their proteins, they're going to have a host cell. Okay, well, this relies entirely on host ribosomes. That's true. Viral protein synthesis occurs inside of the host cell. Well, that's true. Now we got to think about bacteria. Bacteria are living things, right? That means they can reproduce on their own, which means they can copy their DNA, they can transcribe their DNA, they can translate their RNA. Well, if bacteria can do this, that means they have their own ribosomes and they don't need the host. Okay, so, well, bacterial protein synthesis does not occur inside of the host cell? No, it doesn't need the host cell. Okay, the bacteria could be inside the host cell, but protein synthesis is going to be involved inside the bacterial cell. So A is wrong. Okay, so we need to look at C and try to decide between B and C. So it says bacterial protein synthesis relies entirely on host ribosomes. False, right? Bacteria are living things. They have their own. So C is wrong, which leaves B as the correct answer to this question. Good one to know, guys. Good one to know. So in chapter 13.6, we're going to talk about the challenge of antibiotic resistance. So we're going to go over the basic mechanisms of antibiotic resistance. You're going to be able to explain how drug resistance develops and outline some strategies to counteract drug resistance. Okay, guys, see the really big red words? Know these for the exam. You'll be tested on this almost verbatim. Okay, you need to know the 3 basic ways microbes can be resistant to antibiotics. Okay, the first is it's going to prevent the antibiotic from building up inside the cell. Okay, there's 2 ways to do this. You don't let it get into the bacterial cell, or you don't let it stay in. Okay, you get it out as fast as possible. So that's the first one. The second one is you need to be able to prevent the antibiotic from binding to its target. Okay, if you can prevent it by binding to its target, then it can't affect its target, which means it's no longer going to be effective and the cell is now resistant to it. And the third mechanism is we're going to dislodge the antibiotic from its target. Okay, so the antibiotic has bound to it. How do we get it off of its target if we're bacteria? Okay, so these are the 3 ways microbes can be resistant. Know these. Now, in conjunction with those 3 ways, we're going to talk about 6 mechanisms that they can do this. So all 6 of these fall underneath these 3 main ways for antibiotics to be resistant. Okay, this is why antibiotic resistance is such a challenge, is there's a lot of ways for microbes to be resistant. Okay, so strategy number 1, we prevent the antibiotic from staying and accumulating, so building up in the cell. Okay, so there's different ways the bacteria can do this. 1, we can destroy the antibiotic before it even gets in. Okay, right, we just talked about beta-lactamase and how it breaks down beta-lactams. Okay, so it's going to destroy it and make it so it doesn't work anymore. Okay, let's say that the antibiotic does get into the cell. Drug efflux pumps can pump it right out so it doesn't stay and cause damage. Okay, and then maybe our antibiotic is going to use something like porins, so these proteins in the membrane, to gain entrance into the bacterial cell. Well, what if we slightly change the shape of our proteins in the membrane? So they're going to let what they normally let in, but they're not going to be able to let the antibiotic in. So that's going to keep it out as well. So those are 3 of the 6 ways, and they fall underneath that accumulation category. Okay, strategy number 2, we prevent binding. Okay, so the easiest way is to alter the target. Okay, so we're going to have a mutation that changes the target shape enough so that the antibiotic is not effective, but it can still do its job. We talked about this when we talked about the different ways that antibiotics can gain resistant to beta-lactams, right? If we changed the active site of that PBP enzyme, it makes it so that penicillin can't bind to that active site, which means that it's not going to target that anymore, and then cell wall synthesis can keep going. Okay, so that's an example of alter the target. Make it different enough that the antibiotic can't bind, but it can still do its job. Okay, and then second is if we want to prevent it from binding, we need to change the antibiotic shape in order for it not to work. Okay, so we're going to have an enzyme that can recognize these antibiotics, and what they're going to do is they're going to take them and add something like a methyl group. Okay, guys, this is the chemist in Miss Harker. This should be a CH3, not a CH2. Okay, so they're going to add a methyl group, and it's no longer going to be able to bind to its target. Okay, so this is an example of modifying the antibiotic. Okay, so the last mechanism that we're going to talk about is we're going to be able to dislodge the antibiotic once it's bound to its target. Okay, so say that we have a macrolide. Okay, this is an antibiotic that is going to target the ribosome. This is going to target protein synthesis. Okay, so what it's going to do is it's going to bind to the ribosome and essentially shut it off. So what some bacteria have developed are specific proteins for when this happens, that they're going to come in, bind to the ribosome, and they're going to cause a shape change that's going to dissociate or push off the antibiotic so it's no longer causing damage to its target. Okay, this is an example of dislodging the antibiotic. Okay, I would know these 6 mechanisms. Okay, be able to explain what's going on here. Now, we need to talk about the evolution of antibiotic resistance. Now, there are 3 main factors that are going to contribute to antibiotic resistant strains and how prevalent they are. Now, if we look at this graph, this is looking at penicillin resistance and intermediate penicillin. So penicillin intermediates, this means that it's still going to be a little bit susceptible, but it's partially resistant. Okay, so what we can see here, starting in 1985, we're going to see our percentage of clinical isolates, so number of strains that are penicillin intermediate, they're going to increase, and they're going to keep increasing. They might dip for a little bit, but they're going to keep increasing at a pretty normal rate. Okay, these blue bars, this is fully resistant penicillin. Okay, these are drug-resistant forms. Okay, we see that until 1991, we didn't have any penicillin that were, or sorry, any microbes that were penicillin resistant. Okay, but we started to see them pop up in 1991, and we can see that very quickly the number of strains that are going to be resistant to penicillin increases almost exponentially until here in about 2000, 2001, we kind of peak at this 30%. So at that time, penicillin did not work on 30% of the microbes that we were seeing in the clinical lab setting. This is not good. That's a large number. Okay. Now, what we do see is we see the penicillin intermediates. They're going to continue increasing at their own rate in their own pattern. But what we see here is the number of penicillin resistant strains that we're seeing in the hospitals and the labs. It's going to decrease. Now, take a minute, think about it. Why would it be decreasing? Now, some of the factors that led to the evolution of antibiotic resistance for penicillin are the overprescription of antibiotics, right? Penicillin was our first antibiotic. It was prescribed for everything. It was our magic bullet. It was curing things that we didn't think had a cure at that point. Okay, obviously, there's our natural selection example. Patients don't follow the directions. They don't take their full course of antibiotics, so they kill all the strains that are susceptible to penicillin, and leave the ones that are resistant to it. Okay, and the one that we haven't really talked about a lot is the improper prescription of antibiotics. So say you have the flu and you're not feeling good and you want your doctor to do something. There's a lot of patients that go into things like urgent care and different clinics and demand to have antibiotics because they think they'll make them feel better. We all know that antibiotics don't help with viral infections. Okay, they're not going to help against the flu. But a lot of doctors and a lot of PAs, just to get those people out of their hair that just want a drug to take care of everything, they might prescribe them a low dose antibiotic just to get them out of their hair. Okay, so this improper prescription is going to lead to antibiotic resistance just as much as overprescription and patients not following directions. Okay, in fact, sometimes doctors don't want to deal with the patient and they just prescribe an antibiotic and say, see you later. Okay, this is going to all factor into that. Now, you've had a minute to think about these blue lines. What do you think is causing the number of penicillin resistant strains that we're seeing in the hospitals and in the clinics go down? Well, let me tell you, it's the fact that they stopped prescribing penicillin, right? We had all these drugs. We had a 30% clinical isolates number that says that penicillin's not working anymore. Well, if penicillin's not working anymore, we're not going to prescribe it, right? So this is why we see a decrease in the number of penicillin-resistant isolates, so penicillin-resistant strains, than we did in 2000 and 2001, because we just stopped prescribing the drug. Now, with this next example, we've talked about this quite a bit, just not this specific of an example. So we talked about natural selection and how antibiotics can select for antibiotic resistance, for drug resistance. But let's look about what this actually looks like. So we have our patient. They have a form of bacterial pneumonia. Okay, so the strain of bacteria that they catch is susceptible to antibiotic treatment. Okay, that's the blue guys in this picture. Okay, so we get this little bacterium and it's going to divide and multiply, right? Well, remember, bacteria have higher mutation rates, not as high as viruses, but higher than our cells. So what we're going to see is more and more of these blue guys, but every once in a while, we'll have a bacterium that has a mutation that's going to give it antibiotic resistance. Okay, so our infection is going to progress to disease, right? We're going to start to show signs and symptoms. So our patient is going to go to the doctor. Okay, they're going to prescribe an antibiotic. Okay, and it's going to kill all these blue susceptible microbes. Okay, so all the ones that the antibiotic can treat, it kills. Okay, but what's going to happen is this little guy, the one that had that mutation that makes it antibiotic resistant, well, he's going to be left alive and he's going to be allowed to multiply. Okay, so what we do see then is eventually the patient's going to start to recover, right? It worked. We cleared most of the infection, enough of the infection that we no longer have signs or symptoms. Okay, and this is going to allow the resistant strains to grow and develop, and the patient's going to relapse. They're going to get a new strain of this pneumonia that the antibiotics that they're going to be assigned are no longer working. Okay, because even though the antibiotic may still be in the system, it's not going to affect these little guys. Okay, this is natural selection at its finest. Okay, so one of the last things that we're going to talk about in 13.6 is we're going to talk about antivirals. Okay, so antivirals fall underneath the antimicrobial umbrella. Okay, antibacterials, antifungals, antivirals, they're all antimicrobials. Okay, most antivirals are going to target DNA and RNA synthesis. So they're going to be DNA and RNA synthesis inhibitors. Well, we talked about this in that question earlier, is what do viruses rely on in order to replicate and reproduce? Well, they are going to rely on our molecular machinery to help them reproduce. Okay, because antivirals work by inhibiting viral DNA synthesis, that means they're targeting part of our cells. So antivirals aren't prescribed very often. They're only prescribed in very, very serious situations or with the elderly that don't have the immune systems in order to fight a viral infection off. Okay, because the antivirals are going to target DNA synthesis and target our own parts of our own cells, they're going to have very terrible side effects. Okay, because not only are you targeting the virus, you're targeting yourself. You just hope you clear the infection before you cause any serious damage to your own cells. Now, a lot of doctors, when you come in with a viral infection, they're not going to prescribe you anything. They're just going to send you home to sleep it off and have your immune system take care of it. And this is way better than assigning an antiviral with very terrible side effects. Okay, this is why we talk a lot about antibiotics versus antivirals, is because antivirals don't get used a lot. Okay, so that is the end of chapter 13. We have completed all the material for unit 5. We will see you in the next video for unit 6 when we talk about the immune system.